starting back again. We're going to have now a afternoon, evening, nighttime of lightning round presentations and discussion on pathology, genetics, epidemiology, and biomarkers. We're going to start with pathology. After the talks, Dr. Leah Grimberg is going to be the discussion leader. The first talker is Dr. Caitlin Latimer, who comes from the University of Washington. She's going to talk about late NC and the adult changes in thought study. Go ahead, Dr. Latimer.
All right, great. Thanks, Pete. Can you see my screen okay?
Yes, we do.
Okay, great. All right, well, first, I just want to thank the organizers for putting this meeting together and also for the opportunity to get to speak with you all today. I am going to be showing you some of our preliminary data looking at late NC in the adult changes in thought study. And because this really is some new data for us, we only just completed doing late staging across the entire ACT cohort following the 2019 late criteria. So a lot of also what I'll be focusing on is the exciting work that we are planning to do with this now that we have late data in the ACT cohort. So just a brief introduction to the Adult Changes in Thought study. It is a population-based prospective cohort study here in Seattle, Washington. And although the study does not require brain donation to enroll in the study, we do get about 25 to 30% of participants to consent to autopsy. And so our autopsy cohort now has over 800 brains in the biorepository. And so this is a really nice, truly community-based cohort. This slide just demonstrates some of the breadth of pathology that we see in the ACT autopsy cohort. On the graph, each bar is representing an individual brain, and the height of that bar is the neuropath score, which is derived from a cumulative severity score for each of the major pathologies that we look for, with the B score representing tau pathology, C score for amyloid, a Lewy body metric, a microvascular brain injury measure, and then, as I mentioned, now we have late staging across the entire cohort. And I think what a graph like this is really nice to show is, as has been discussed earlier this morning and has come out in the literature, there is a lot more late pathology in the demented subjects than those without dementia. So TDP-43 definitely seems to be a strong influencer of dementia. But we wanted to do, now that we have this data, is to start asking some deeper questions about that, but to first make sure that what we're seeing in the ACT cohort matches what's coming out of a lot of the other cohorts. So we first asked the question whether or not, and to what extent really, does the late pathology contribute to the risk for dementia in this cohort? And again, the data really reflect what has been found in a lot of these other cohorts, and that if you have Alzheimer's disease pathology alone or late pathology alone, you do have an increased risk for dementia, with it being slightly stronger for the Alzheimer's pathology. But what's really impressive is this combination of late NC and Alzheimer's pathology that really increases the risk of dementia. And so this core diagram on the right here also illustrates that point nicely, showing that in the Alzheimer's disease only, you get about 2/3 of folks that are segregating to dementia, and 1/3 are maintaining cognitive function. And that's in contrast to the ADNC plus late NC group, where really just about everybody is getting pushed to dementia, and only a very few are able to remain dementia free. And so this has been a concept that I've been really interested in, is why do we see this really strong association with Alzheimer's pathology and late NC, as was coming up earlier in the talks this morning. And so to look and see if this is merely just an additive effect, or is there potentially some synergistic effect between Alzheimer's pathology and TDP-43, we started looking at quantitative metrics of some of these pathologic proteins across multiple brain regions throughout our ACT cohort. And we did not see any relationship with amyloid burden in cortex, but we do see a relationship between neocortical tau pathology, the burden of tau as measured on using digital slide analysis techniques. And this was particularly true in the middle frontal gyrus, where there was an interaction between latency, tau burden, and the risk for dementia. And so this does at least lend some support for this thought that perhaps there is some synergy or relationship between TDP-43 pathology and Alzheimer's pathology, particularly tau. But this is really the tip of the iceberg. There is a lot of work to do in this cohort. And so we are going to continue looking at quantitative assessments of ADRD throughout the brain in the ACT cohort. This will include some duplex staining to further delve into this potential relationship between tau and TDP-43, also employing some artificial intelligence to look for patterns that may escape just the human eye. And then finally, there are a number of genetic studies that are aimed at trying to get at some of the genetic underpinnings between late NC, but also potentially this pleiotropy between tau and TDP-43, with the goal of identifying genetic modifiers that we can then test in model systems to hopefully identify potential therapeutic targets. And so with that, I just want to thank all the people that have made generating this data possible, particularly the donors and their families. I won't go into everyone here because I think my time is probably up, but it clearly takes a village. So thank you.
Thanks so much, Dr. Latimer. Wonderful talk. Next speaker is going to be Dr. Matt Sykowski from Houston Methodist Hospital, who's going to talk about early late patterns and associations of TDP-43 pathology in the amygdala region. Take it away, Dr. Sykowski.
All right. Thanks, Dr. Schneider. Thanks, Dr. Silverberg. Thanks, Dr. Nelson, for the invitation to speak. So this is some work I've been doing looking at the amygdala and patterns of TDP pathology and aging or late. So TDP pathology, as you've heard today, is extremely common at autopsy. These are commonly the settings in which we see TDP pathology, as we heard about earlier: Alzheimer's disease, Lewy body disease, what's been termed hippocampal sclerosis, dementia, as Dennis mentioned, the aging brain or pathologic aging. And so for me and my colleagues here, the idea of having reporting TDP plus or minus hippocampal sclerosis is extremely useful. So aside from the radiologic, genetic, and clinical features, which you've heard about, for me as an autopsy person, it's a very helpful unifying concept that this idea of TDP and hippocampal plus or minus hippocampal sclerosis in these various settings could be called late. So in terms of where we look for late when we do autopsy pathology, stage 1 has been described today is in the amygdala, the medial temporal lobe. Stage 2 has been described in the hippocampus, and stage 3 in the frontal cortex. And there's more nuanced and extended staging schemes. I think this is generally a pretty useful one. This is not the only one, but this is a 3-stage classification system that's pretty well accepted. One of the problems for me as someone looking at these is the amygdala is a very complex structure. So when I hear amygdala stage 1, I don't really know exactly what that means. And you see on the average MNI brain here, that box bounding the amygdala, but in reality, this is what amygdala looks like. So very heterogeneous structure, even within the amygdala proper, great variation in the cytoarchitectural detail. The entorhinal cortex is adjacent. The basal forebrain is merging with the dorsal part of the amygdala. The claustrum and insula are very close by. The ventral putamen is very close by. So you have a lot of different structures all coming together within what's bounded in this box in the MRI. So one of the things that we wanted to look at was, where is this pathology beginning and what might that tell us about what late is and help us better classify it. So we looked at 184 samples with representative anatomy of the amygdala region. This is a collaborative study between Kentucky and Houston. The patients we looked at had a median age of 85 years at death. So we specifically were looking at much older patients. These patients were cognitively normal or were affected by Alzheimer's disease or Alzheimer's disease-related dementias. We did not look at FTLD, ALS, PSP patients. The samples had a median Brock stage of 3, and most of the samples, the majority, were somewhere in the no, low, or intermediate ADNC range. So this is not a study of high ADNC with TDP. All of the samples we looked at were with H&E, LFB for myelin, Nissl for cellularity, and structures like entorhinal cortex, and we used both phospho and N-terminal TDP antibodies. I think importantly, we looked at the TDP blinded to any of the global neuropathologic and clinical diagnoses, as well as the risk alleles. And the risk alleles that were studied include those listed here, which have all been described in some form as being a risk factor for TDP. So In our study, amygdala region, this is a schematic showing some of the structures that can be seen in the amygdala region. The largest part of the amygdala region, when we look at a slide, the amygdala region is the basolateral nuclear group by volume. It's going to be the largest part of the amygdala. The superficial part and near the peel surface is the cortical-like region. There's another lateral division, which I'll show you in a second, probably here, that is actually quite easy to pick up even on an H&E stain. And then below the amygdala, we have the entorhinal cortex, the transentorhinal cortex on the bank of the collateral sulcus. And opposite of that, we have neocortical cortex, which is occipitotemporal cortex. So take home number one from our study is that early late involves the amygdala region, not just the amygdala. So when we say stage one, we should probably refer to amygdala region. And this heat map is a bit complex, but the big picture thing here is, 3 of the columns are actually amygdala proper. They're subnuclei within the amygdala, the cortical medial, basolateral, and then the lateral division, the lateral nucleus. Everything else you're seeing there is not amygdala. And it's not hard to, if we look at enough samples, to find cases where we can see pathology in structures that are not amygdala when amygdala itself has pretty limited involvement. The second take-home point is that early-late in the amygdala region is heterogeneous. There is no single pattern that we can find in everybody. So this is a sort of a simplified map of where we might see these types of pathology. So we can see round NCIs, particularly in the amygdala. We can see the sort of, as Dr. Newman alluded to earlier, the FTLD type A-like pathology with small neurons having small neuronal cytoplasmic inclusions and adjacent neurites in lamina 2, particularly around the collateral sulcus, either side of the collateral sulcus. Occipitotemporal cortex is a very good place to see that. We can see flame-shaped type TDP inclusions. They look very fibrillary, almost tau-like, so called these NFT-like NCIs, and Dr. Dixon and Dr. Joseph have called that type beta, and especially, as Pete mentioned earlier, where you see Alzheimer pathology, that's a very common type of place to see that form of TDP. We can also see, in some cases, it's not common, but you can see granular pre-inclusion pathology, actually first described by Dr. Trojanowski in a paper about ALS and FTLD. It's a very useful finding in ALS in particular. We see this in a lot of ALS brains in this region and other regions, but you can see this in late. We see thick neurites, almost resembling like a Lewy-type neurite, particularly in amygdala. And then we can see subpeel and white matter processes, and especially overlying the cortical-like nucleus in the amygdala and in the white matter that's between the ventricle and the neuronal cortex. So these are the different heterogeneous type of structures that you can see in late patients. So the next idea was to take those patterns and just using the TDP to cluster these using a kind of an unbiased approach, a hierarchical clustering algorithm. And that gives us 5 patterns of TDP based on the combination of the things that you can see within cases. So the brown dots are where the feature is present, and present on both the phospho and the non-phospho or N-terminal TDP. And the gray dot is where the feature was absent. So for instance, you can see areas with predominantly an NFT-like inclusion pathology pattern. We can see groups of cases with round NCIs in the amygdala and neurites. We can see a large number of cases with non-NCIs and predominantly a process-predominant form of TDP. And on the right end, we can see cases that don't have all of the neurofibrillary type pathology, but have a lot of lamina 2 and granular pre-inclusion pathology. So one of the big pictures of doing that then is that meaningful? Well, if we go backwards to the data, which we're blinded to when we do that classification, it actually does appear to be meaningful.
We do got to kind of speed up a little bit because I want to get everybody in.
Okay, so I'll take questions about this offline, but real briefly, basically the staging of the cases is different based on the clusters. The amount of Alzheimer's disease neuropathology is different based on which group you're in. There's one group, particularly pattern 4, that's very common in patients with diffuse and limbic Lewy body disease. The frequency of hippocampal sclerosis differs based on which group you're in. Interestingly, the E4 allele differs which group you're in, and especially the beta, it's way overrepresented in the cluster 2. So I think I've lost control of my slides, so I'll stop there. Oh, sorry, summary. So number 1, summary point, early late involves the amygdala region, not just the amygdala. Number 2, early late in the amygdala region is heterogeneous, but the foci are fairly predictable as it progresses. Number 3, there are several patterns that emerge. Number 4, these patterns differ in their pathologic, clinical, and genetic associations. And not mentioned in the prior slide, but the TMEM106B risk allele strongly associates across all forms of late in the amygdala, which is pretty remarkable considering the heterogeneity of what I showed you. So thanks for the time, and there's my email if anybody wants to reach out. Thank you.
Wonderful, wonderful. Great talk, Dr. Psychowski. Sorry to cut you off. So the next person is going to be Dr. Margaret Flanagan from Northwestern University. Her topic is synaptic changes in the setting of coexisting TDP-43 and Alzheimer's disease neuropathologic change. Take it away, Dr. Flanagan.
Thank you so much. Could you please stop sharing your screen and then I can share mine? Oh, perfect, thanks. Great, so thank you very much for hosting this event today and for specifically inviting me to present. I'm very excited to share quickly some of the most exciting preliminary results from a study that has been looking at the entire spectrum of Alzheimer's disease and neuropathologic change with and without TDP-43. So as everybody here already knows, it has been described in multiple different cohorts, Rush, as well as by Pete's group, the Mayo group, as well as University of Washington by Dr. Latimer, and the ACT cohort. And in the NUN study and Honolulu Asia Aging study, which I primarily had been working with, we also found that there were lower cognitive test scores observed among those with pathologic TDP-43 in both cohorts. But this just raises the question, what are the specific TDP-43 associated disease mechanisms that may result in cognitive impairment and ultimately different types of dementias? And so I'm doing a large study and just showing these 5 groups here just for some brief comparisons today. And so we're basically looking at a control group, not cognitively impaired, that's low Alzheimer's without TDP, and that compares to the group with the non-amnestic dementia behavioral variant frontotemporal dementia, which has positive abnormal TDP-43. And then for groups 2 through 4, it's interesting because they all have high Alzheimer's disease neuropathologic change that were matched. One of the groups, the amnestic dementia probable Alzheimer's disease dementia group, had a late presence. So group 3 is our late group specifically. And it's quite interesting because neuropathologically, both groups 2 and 4 have high Alzheimer's disease neuropathologic change and lack pathologic TDP-43. And as you can see in the group that has the highest level of plaques and tangles, as well as late TDP-43 pathology, we actually found doing multiplex immunofluorescence studies for different cell-specific markers and different types of TDP-43 antibodies that we had the lowest number of astrocytes seen in the late group. And interestingly though, there's a difference when we look at AMPA receptor staining using immunohistochemical staining, and we looked here just using optical density. We found that there was a difference between groups 2 and 3, but group number 4 that has a different clinical phenotype did not show a difference from our late group number 3. So we wanted to take another look at things on a deeper level, and I'm just going to be briefly talking today about the presence of matched tau, so Brock-Tingle stage 2 lacking amyloid, and comparing TDP-43 pathology present and absent. I have a lot of interesting results for just amyloid as well, and then adding the 2, but for the sake of time, we're just sticking to tau and TDP. And so essentially we used digital spatial profiling with nanostring to look at different specific proteins of interest. For this, we wanted to look at the synaptic panel. And what we did was we selected multiple different areas to look at. And in addition to that, we masked to look at different specific cell types that included microglia, neurons, and astrocytes. For those of you that are not familiar with this technology, briefly, you just take formalin-fixed paraffin embedded sections, you place them onto a glass slide as usual, label it with your probe mix, and from here you are able to cleave the tags and quantify the proteins to map back to the tissue, which gives you spatial context in addition to the quantitative measurements. From here, you do all of your QA, and you can do different volcano plots. So we specifically wanted to look at, again, the synaptic markers to see if we can find any hits specifically for Alzheimer's disease, specifically only tau with TDP-43. And what we found, in summary, is a really interesting difference. So that when we matched the cases, so first, we just want to look at tau positive versus tau negative. And what we saw here, in cases were matched for age and sex, and all coexisting neuropathologies were excluded other than those I mentioned. So no alpha-synuclein. There's no APOE4 allele present in the case stratification. We found that actually in the tau positive group, there was significantly higher vacuolar protein sorting ortholog 35 or VPS 35 in the new and masked neuronal population specifically. In addition to this, when we then looked at our comparisons and wanted to look at groups that were matched, Brock stage 2 for tangles, TDP positive, which was impaired clinically, so dementia, versus cognitively intact for the TDP negative and tau by itself, we saw an inverse relationship, which is quite interesting. And just to give everybody a brief summary of what this VPS 35 does. So basically it's related to autophagy, and this diagram here on the right hand side shows that in healthy conditions, the orange right here, when the VPSs of normal levels are sufficient and the AMPA receptors are able to be present and localized at the synapse. The mitochondria are stable and you don't have any neuronal degeneration. However, it's been specifically described in the setting of Parkinson's disease that the VPS 35 levels are low, or if you mutate them mechanistically, the AMPA receptors become absent from the synapse and mitochondrial homeostasis is disrupted, resulting in neuronal degeneration, which is facilitated by an abnormal protein aggregation, as I mentioned, which has been previously described in alpha-synuclein. But this perhaps may also be the case for TDP43, at least in the setting of coexisting tau pathology by itself. And that's it. I'd just like to thank my lab, the Meslem Center, the Department of Pathology, all my collaborators, and of course, the participants themselves. Thanks.
Great talk. Thank you so much, Dr. Flanagan. Next talker is Dr. Melissa Murray from the Mayo Clinic in Jacksonville. Her title of her talk is "The Variable Effect of Latency on Cognitive Decline in the Flame Series." Thank you, Dr. Murray.
Thank you. And thank you for the opportunity to present our work today. It's been already a very interesting day. And some of what we've discussed hopefully aligns within the Flame series. So for the purposes of generalizability, this is an autopsy series that was derived from memory disorder clinic patients across the state of Florida, all of whom at specific memory disorder clinics have the option to elect for autopsy as a free service. And this would not be possible without many individuals contributing, especially Dr. Dwara, Greg Cousteau, Graf Radford, and Dixon. And so really just to kind of get into what we were interested in, this study kind of derives from when we're really designing studies and the differences that may evolve when you're looking at the underlying molecular aspects between Alzheimer's disease that's pure versus those with coexisting pathologies. And so looking in our series, we separated out Alzheimer's disease cases with concurrent amygdala-predominant Lewy bodies, hippocampal sclerosis of a TDP43 etiology, and Lewy body disease. But even still, what is pure AD? How many more would we need to cut if looking at those with late NC, so outside the context of hippocampal sclerosis, as we've discussed today, stage 1 or above, and what do we do about cases with cerebrovascular disease? And so I won't get into too many details in our lightning round, but we really need to be aware of what may be contributing. And at least here from the context of cognitive decline, it's great to be able to talk in the context or following Dr. Boyle's presentation, because how we've stratified was really inspired by the work of her group from Rush. Namely is when we think about cognitive decline, we really do need to think about the decline that occurs near death and that occurs prior to. And so stratifying by less than or equal to 5 years and looking at the MMSE scores and looking then substantially at the decline, we find that individuals in the Flame series decline at about 9 points per year. So this is out of a total of 30 in comparison to the 3 points per year lost earlier in the disease course. So it was nicely confirming prior results. in this large series of autopsy-confirmed Alzheimer's disease cases, we actually, when controlling for these pathologies, don't see a specific contribution from Brock and Thal in this series. We also don't see a difference. So kind of once again, reminding you earlier in the cognitive decline process or nearer to death, we don't see a specific contribution from amygdala-predominant Lewy bodies, and I'm not quite surprised by this. Really, when we think about ALB, we think about a Lewy body variant of Alzheimer's, really in the context of an extensive and advanced Alzheimer's case. However, when we look at Lewy body disease, so this could be brainstem Lewy body disease, transitional or diffuse, an Alzheimer's disease case with Lewy body disease, we found earlier involvement and contribution to cognitive decline that was not observed nearer to death. Specifically looking at the hippocampal sclerosis cases who were defined by their disproportionate nature of TDP-43 in the hippocampus outside the context of tau pathology, we also see a contribution to the decline of the cognitive status in the series early, but not near the time of death. Interestingly, and I think we would also have come to consider this, and this is part of the discussion we've had today, what do you do with late NC in the context of hippocampal sclerosis? So for this model specifically, we removed the cases with concurrent hippocampal sclerosis, or how we've defined it, and looked specifically at the individuals with late NC, stage 1 or greater, who actually were found to also have Alzheimer's disease. And once again, we find that the cognitive decline or the contribution may actually be occurring earlier than later. We don't necessarily in this series see a contribution of cerebrovascular disease, but we do see a contribution of age onset. And I'm actually joined today by Dr. Grinberg, and we recently were awarded a young onset AD grant. And so I want you to pay specific attention to this because a lot of us study late onset forms of dementia. And what this data is supporting, and it's something that Dr. Schneider brought up earlier, is that our older patients may not have as severe of a form of the disease. And so we didn't see that dramatic decline, or we don't even see a contribution. So the older individuals do not have as low of an MMSE as the younger individuals in this series. And when controlled for, we don't see a contribution in this context between sex and education. Thank you for this opportunity. And I appreciate all of my grant support and collaborators. Thank you.
Great. So with that, we segue to Dr. Grinberg, and I won't say a thing.
So thank you very much. We had now 4 talks on neuropathological aspects of TDP-43, and a common theme of all these talks is that TDP proteinopathy is much more heterogeneous than ones that we appreciated before. And one of the reasons that we know that it's because of all these deep phenotyping studies that are being done in neuropathology. Neuropathology evolved much beyond we sitting under a microscope and doing some diagnosis to really interrogating if what we are seeing really goes aligned with the criteria that we have and how we transform it in new knowledge. Also, a common theme of these talks were that the tools to examine post-mortem human tissue had evolved a lot in the past years. We are seeing in situ proteomics. We are seeing deep phenotyping with the molecular tags in situ, and I'm really looking forward for what will happen in the future. We only have 2 minutes more for this discussion. We won't be able to have a question and answer session live, but I encourage everybody to add your questions and your comments in the Q&A because the speakers have the opportunity to write the answer. So let's not miss this opportunity here. I think with this, I mean, we are about at the hour. And I think I will hand it over to you, Pete, again.
Thanks, and apologies to all of you all for short shrifting you. We had 4 panelists. The other 3 sessions only have 3. I'm sorry to both get in 4 outstanding talks and to short shrift us for having Dr. Grinberg's input, but I hope that we can do a side chat and or QA discussion about these wonderful talks that each of you all gave as we move on to the next step, which is about genetics. So again, I apologize to everybody. We put in 4 instead of 3. The other 3 sessions have 3 talks each. please do ask questions for Dr. Latimer, Dr. Sikowski, Dr. Flanagan, and Dr. Murray in the Q&A and in the chat. I think they all have fantastic short presentations, and we will go now on to genetics. Anybody else have anything they want to say? Dr. Schneider? Okay, so Dr. DeJager is going to be our discussion leader. We hopefully will get some more actual discussion this time. And our first talker is Dr. Dave Fardo from the University of Kentucky. He is going to describe candidate genes for limbic-predominant age-related TDP-43 encephalopathy.
Great. Thank you so much to the organizers for putting this meeting together and for the opportunity to present our research. I'll be presenting analyses from a couple of our group's recent papers on the genetic susceptibility to late. The first set of analyses we'll cover examines candidate genes for late neuropathologic changes in hippocampal sclerosis in an attempt to generate hypotheses about how and why late NC can transition to comorbid hippocampal sclerosis. Then we'll investigate data from a novel late NC risk gene, WOX, that has been implicated in multiple neurodegenerative diseases. The first candidate genes for hippocampal sclerosis, as we've heard before, came from frontotemporal lobar degeneration. Most common cause of FTLD TDP is mutation in the granulin precursor or progranulin gene. Then variants in TMEM106B were discovered via genome-wide association study, or GWAS, of FTLD TDP. After those candidates were confirmed to associate with hippocampal sclerosis, the first GWAS specifically for HS was conducted in 2014 by our group and found variants in the ABCC9 gene to be genome-wide significant for hippocampal sclerosis. A later GWAS for multiple neuropathologic endophenotypes found KCN-NB2 to be suggestively associated with HS. APOE findings have been mixed, but we examined it due to its ubiquity in ADRD research. While exploring these genetic susceptibility factors, we wanted to think about what predisposes some individuals with late NC to also harbor comorbid HS, has been discussed earlier this afternoon. In our first study, led by Dr. Adam Dugan, we used genetic and neuropathologic data from two combined sources. First, the religious order study in the Rush Memory and Aging Project, ROSMAP. And second, NAC participants mapped to GWAS data from the Alzheimer's Disease Genetics Consortium, the ADGC. In total, we were able to phenotype over 1,400 research participants, and we're curious about how these genetic risk factors relate to one or a combination of these pathological diagnoses. Particularly, although the data sources represent multiple study designs, we noticed the rarity of HS alone and wanted to generate testable hypotheses regarding the relationship between late NC alone and late NC with comorbid HS. We can't go over all analyses in this quick talk, so I encourage you to read the paper for more details. We found a variety of associations and focused on an attempt to adjust out the potential effects of the second pathology. Overall, we find robust association between late NC and the granulin, TMEM106B, and APOE genes, as well as associations between HS and the ABCC9 and granulin genes. These results lead to the hypothesis that the genetic correlates of progression to late NC differ from those of late NC with comorbid HS, as you can see drawn in figure C. We also looked at WOX as a novel late gene. GWAS requires tens of thousands of subjects and can often rely on phenotypes subject to various forms of misclassification. For example, recalled family history of AD as a proxy outcome. When the Kunkel et al. AD GWAS came out a couple of years ago, we were reminded of analyses from our 2014 HS GWAS that included a variant in WOX, as well as a long-standing hypothesis that some AD genetic hits could in fact be driven by AD mimics. Again, led by Dr. Dugan with the data described earlier, We examined the WOX-MAF locus for neuropathologic endophenotypes, and after adjusting for multiple comparisons, found variants associated with late NC, HS, and brain arteriolosclerosis, but not with AD neuropathologic changes. Again, I refer you to the paper for more detail. In summary, we find genetic associations are consistent with the pathological model, such that the genetic risks for transition to late NC differ from those for transition to HS. We also find the WOX gene appears to be a risk factor for late NC rather than AD neuropathologic change. This bolsters the importance of late NC, as well as for neuropathologic endophenotypes in general, and gives credence to the hypothesis that some clinical AD hits are driven by other phenomena. Thank you.
Thank you very kindly, Dr. Fardo. And wonderful talk. The next person is Dr. Hyun-Sik Yang from the Harvard University, whose talk is going to be about the shared and distinct genetic risk factors of late NC. Take it away, Dr. Yang.
Okay, thank you very much for the opportunity to share what we've learned about late NC using genetic approaches. So, interestingly, Alzheimer's disease and late NC has a very close association, and it's unclear why. And natural question that we can ask is, do they have shared genetic risk factors and shared molecular programs that govern both of their pathophysiology? In fact, APOE4, the strongest genetic risk factor for late onset Alzheimer's disease, is also associated with late NC, and with an odds ratio of 2.0 for higher late NC stages. And that association persists even if we control for a quantitative amyloid tau burden from ROS-MAP study. So it seems that APOE4 has a pleiotropic effect on both AD pathology and late NC. And interestingly, when we did some modeling afterwards, APOE4 had an association with hippocampal sclerosis, but that association was mostly mediated through late NC. So at least a portion of hippocampal sclerosis seems to be downstream of late NC from the genetic evidence. On the other hand, strong association between AD pathology and late NC could not be completely explained by APOE4 alone. So with APOE4 as a shared genetic risk factor, we looked into another genetic risk factor of late NC, TMM106B SNP. That is initially discovered as a genome-wide significant risk factor for FTLD TDP, but later turned out to be also associated with late NC. So interestingly, this SNP was associated with large-scale gene expression changes in ROS-MAP, dorsolateral prefrontal cortex RNA sequencing data. And it regulated multiple gene co-expression network, including the one we call module 110, which is enriched in genes that have important roles in lysosomal regulation, as well as myelination pathways. And interestingly, that particular gene co-expression module also had positive correlation with late NC burden, as well as amyloid beta burden, but not with tau. So after a series of mediation modeling, this is the pathway diagram that we landed on. So here, TDP43, TMM106B risk allele is going down to module 110 and then increase module 110 level, whereas APOE4, a beta pathway goes down in conversion to module 110 and increase that module's expression. And then in turn, module 110 is associated with late NC and is a likely upstream for our modeling. Of course, there is still unexplained direct link between amyloid and late NC, even after considering for module 110, but this now provides us an interesting shared molecular program between late and amyloid beta. So this is a model that we had after doing this study. So both amyloid beta and TMM106B, as well as granulin variants, lead to the dysregulated lysosomal autophagy pathways. And then in turn, this is leading down to late NC, and that probably downstream of that, what happens is splicing dysregulation and neural dysfunction that leads to clinical syndrome. So these are all possible scenarios, not like definitive, but using multiple layers of genetic and transcriptomic data, we were able to get to the shared molecular links between Alzheimer's disease as well and late. Finally, to look into the distinct genetic risk factors of late that are not necessarily shared with Alzheimer's disease and FTLD, we looked into genomide association study. This is a preliminary unpublished data from 1,300 people from ROS-map cohort. And there's one genomide hit, which is APOE epsilon 4. The exact same SNP that is used to define epsilon 4 is the single highest hit there. And when we looked up previously described genetic risk factors shared with FTLD, TMM106B SNP, as well as granulin SNP, they also had nominal significant association with latency, although not as strong as APOE epsilon 4 and not the strongest besides APOE. But interestingly, although did not reach genomide significance, there were several interesting hits that reached our pre-specified suggestive threshold of 10 to the minus 5. And those genes, interesting enough, were enriched in synaptic neuronal genes. And also interferon alpha cluster came up, and other genes in type 1 interferon pathway also showed up here. So these results together shows interesting shared genetic risk factors between latency with AD as well as FTLD TDP, and suggests that there may be some distinct genetic risk factors leading to latency in a unique way. So with that, I would like to appreciate all the ROS-map study participants and MPAD staffs, as well as mentors and collaborators across many institutions. Thank you.
Thanks kindly, Dr. Yang, for excellent talk. Next speaker is Dr. Eddie Lee from the University of Pennsylvania, who will talk about late genetics, interactions, and outliers. Take it away, Dr. Lee.
Great. Is my slide showing appropriately?
Yes, sir.
Okay. First, I want to thank everybody. The outpouring of support has been amazing. It's remarkable that it comes in waves. As Europe wakes up, we get love, and as Asia wakes up, we're getting condolences. And so everybody at Penn appreciates the thoughts. And I've taken some liberties here for this presentation, and I hope you bear with me. And so we started looking at this in this paper several years ago, just looking at our brains at the CNDR UPenn Brain Bank. This initial study was about 766 autopsies. Of course, this is a tertiary referral center. These are very specialized cases. But we wanted to look at the extent of comorbid neuropathologies in our cases. And here you can see our different categories. This is our minimal pathology group, meaning not Alzheimer's, doesn't meet criteria for anything. Intermediate high Alzheimer's, tauopathies, and Lewy body diseases. And you can see that TDP-43 proteinopathy is seen in the setting of Alzheimer's disease and the tauopathies, but not all of them, and also in the Lewy body diseases. And so incredibly prevalent, highly impactful in our brain bank. And you know, our center and many others have looked at this. This is comorbidities in the setting of intermediate or high level of Alzheimer's disease neuropathologic change. And you see that a high proportion of these cases indeed have TDP-43 proteinopathy. So how are these kind of studies done? So this is the faculty picture for the Department of Pathology in 1981. And this gentleman here is Nick Ganatis, and he had the foresight to recruit and have this individual here, John Trojanowski, become faculty. And so he became faculty in 1981, and quickly within a few years, getting his feet steady, he started banking tissue and created the brain bank. And so this is one of our first cases. This is our 6th ever Alzheimer's disease case, a 76-year-old Black man with a diagnosis of Alzheimer's disease, diet of infections. I love that the workup is about 4 sections with H&E, Bodian, and thioflavin S. But this was the state of the art. And he did diagnose senile dementia of Alzheimer's type. But I do know that in the note, you could also look at, you know, already back in the 1980s, commenting on the comorbid pathologies being Lewy bodies, mild pigmentary incontinence, and a few globose tangles, which of course back then we did not understand what they meant. And so this was the Cacciatarian era. You know, this is even before CRAD for the younger people on this symposium. Of course, John was instrumental in updating this to incorporate both amyloid and tau in the NIA Reagan criteria and the more recent NIA Alzheimer's Association criteria. So here's my liberties. I just want to highlight some of the seminal work that has been done with the tissues from our brain bank. And so with his longtime collaborator and life partner, Virginia, this is a paper in Science in 1991. At the time, there were suggestions that tau was the protein that made up paired helical filaments in neurofibrillary tangles. And this is kind of the nail that solidified this. Interestingly, when this paper came out, they did get correspondences from others in the field saying, we think you're wrong. I don't think it's tau. But you can see these beautiful filaments here. And for the people who have not read this paper, this is not an immunoblot. This here is a pure Kumasi stain gel. This is the pure protein tau shown conclusively that this is forming neurofibrillary tangles. 1997, together with Maria Spillantini, this is the first time that Lewy bodies were shown to have alpha-synuclein. And so these are images of dementia with Lewy bodies, cortical Lewy body pathology, which had never been seen before. And then fast forward in science in 2006, again, with Virginia Lee and Manuela Newman, who presented earlier today, showing that TDP-43 forms inclusions in ALS and frontotemporal dementia. And this, of course, being very important for this conference as the basis for late neuropathologic change. So John was an amazing champion of pathology. And this, I just wanted to highlight these 3 quotes. 2 of these quotes were from the 1900s. At the turn of the century. One of these is from 2000, and just talking about how the near end of the century that the CPC was almost done, that research should not be done on the dead house of pathology, but rather physiology. This is the one quote that was from around 2000, describing whether or not TDP-43 matters, calling it potentially superfluous junk, that it's pathology, but perhaps not pathophysiology. And so I applaud John for sticking it out and championing pathology. And so if we revisit that case, we could call it, you know, we know they had Alzheimer's type dementia, but we know that they also had, sorry, there's a typo here, an intermediate level of AD change that should be a B2, but also had late neuropathologic change in Lewy body disease, brainstem predominant. And so we spent years backfilling all of our cases. And so this is 1986 and perhaps one of the earliest cases of known late neuropathologic change. So more specific for our work today, I want to highlight work done by Dr. Maiko Umera, and she has a poster presentation at this conference. And so with this backfill of all of our cases, we've looked at nearly 1,000 cases that are fully neuropathologically phenotyped into different groups, either being aging, where they don't quite fulfill full criteria for Alzheimer's disease, AD only, mainly Lewy bodies, or a mix. And you should listen to her talk because she has a lot of clinical and other data in there. The high level results is that late neuropathologic change is associated with age, as everybody has seen, is also associated with ADNC, meaning the more Alzheimer's disease neuropathologic change you have, the greater your risk for late neuropathologic change. And importantly, late is also associated with LBD subtype. So neocortical Lewy body disease has a higher incidence of late compared to transitional, which is higher compared to brainstem predominant. And we could stage late neuropathologic change in the setting of Lewy body disease. And so here's just looking at the prevalence across stages and the statistics for the conditional probability analysis. But just to give you a high level analysis again, in the hippocampus, there are interesting and perhaps subtle changes. For example, the dentate gyrus is involved earlier in Lewy body disease. Interestingly, the brainstem is affected much earlier in Lewy body disease than it is in Alzheimer's disease, where the brainstem is really, really late phenomenon. So this tells us, and again, for the hippocampal stuff, I urge you to go look at Maiko's presentation. So this shows us that there are interactions between these 3 main neuropathologic drivers that John and Virginia and Manuela and others have discovered. I put A beta in parentheses here because we do think A beta relates to tau, but in my mind, tau, TDP, and alpha-synuclein are the 3 henchmen that really cause neurodegeneration. And figuring out how these pathologies interact with the genetics, I think, is going to be a challenge. And so, for example, in our cohort, we do see associations with TMEM106B genotypes and granulin phenotypes, genotypes. We did not reach statistical significance for APOE. Again, these are relatively small for a genetic study. And so determining whether risk factors are primarily driving Alzheimer's disease tau versus, driving TDP-43. We did not get significance for genetic markers associated with alpha-synucleinopathy, but I think these studies will require larger cohorts for us to analyze. And then one final study that we're working on publishing, this is Alexandra Young and Jake Vogel, together with John and Oscar, and they have a presentation as well that you could look at. Again, so they use a sustained model. So this is a statistical model. You may remember there's a Nature Medicine paper recently using this with Jake Vogel and Alexandra, where they looked at neuroimaging metrics and were able to define 4 different subtypes of Alzheimer's disease and define their progression. So using that exact same statistics, we turn this to our brain bank to look at FTLD, TDP, ALS, and late. And you can see here that the precision and recall is astronomically high, 95 and 96. And so this is kind of what the data looks like, where on the diagonal, what we thought neuropathologically was predicted by their sustained model. Again, this is completely data-driven analysis, and there are a few outliers. And so overall, this is telling us that indeed FTLD TDP looks in our data set looks distinct from late, and in almost all cases that we are able to categorize these things. Importantly, the outliers are truly strange cases. And so there are a bunch that were, do we call it FTLD more than ALS versus vice versa? I want to point out that this is highly enriched for genetic variants, including 2 C9 cases, 2 TBK1 cases, and this is out of 4 in the entire brain bank. 1 NIPA1 variant, the only one that we could identify in case, a TREM2 variant, and a taxon2 intermediate expansion. And then even weirder things like PSP, which with what looks like some late PSP with some ALS or with motor neuron disease as well. And then this case where every slide showed everything with TDP, AD, hippocampal sclerosis, CT, and Lewy body disease. So it's not unexpected that the extremely weird cases cannot be categorized neatly, but really those are truly the outliers, which kind of proves the rule in our database. I added this slide just because people are talking about biomarkers. I think the hope there lies with molecular genetics. This is a study where we took human brain tissue and we could sort them for fact sorting. And so this is what normal brain looks like with neurons that are TDP positive and glial cells. In FTLD TDP, you see you get this second population. And so these are neurons that have lost their TDP43. And we could sequence these and do deep molecular phenotyping. And there are thousands of genes that are altered, right? So I think the hope for a biomarker does not necessarily lie in neuroimaging. I think the amount of pathology we see is going to make a PET tracer difficult. I'm not saying it's possible, but very difficult. I think the structural MRI studies will be sensitive to TDP43, but not necessarily specific. And so I think looking at the functional defect of TDP43, where these genes are massively dysregulated, has hope for a fluid biomarker looking for these downstream proteins. And we have a tenure track position open for fluid biomarkers if anybody is interested. And I'll share this data set with anybody who's interested. And so some conclusions. I'll say that since this is an interactome map from Corey McMillan, who's a colleague in our FTD center. This is mapping all the publications on TDP43 since 2006 from over 11,000 authors. And each author is a node here. And I just want to highlight John Trojanowski being the yellow dot in the center of this universe. And so his productivity is remarkable, an H index of 246. Tony Fauci doesn't reach John Trojanowski's H index almost, but close. And so I just started pulling in images of the faculty at Penn who have been directly impacted by him, funded through our Alzheimer's Disease Center, FTD Center, our Lewy body disease program. This is me when I defended my thesis with Virginia and John. And again, I appreciate everybody's support.
Thanks for a great talk, Eddie, and it's great to be reminded about John, and what a great loss. Okay, Dr. Dieger, you want to take it away?
Certainly, and again, 3 great talks, and thank you, Dr. for this, or these nice memories of John. So to get us started, I want to go back to Dr. Fardo's presentation. And again, I think this is sort of one of the main topics we're talking about today is sort of this heterogeneity and the combinations of different pathologies that we see. And so you propose, again, that certain variants may be more involved in one or the other of the pathologies. But of course, this is limited by the power of the current data sets. And so how confident are you that these are really sort of maybe uniquely attached to a particular manifestation, or that perhaps we just don't have the power yet to really exclude the possibility that they're more widely involved?
That's a great question. I mean, I don't think with 1,400 some subjects that we can be completely confident that any one of the differential effects of genes are for one pathology or the other. So I mean, ideally, we would have, even beyond the neuropath data, we would have longitudinal measures, which is not really a possibility with autopsy-based phenotypes. So, I think to your point that it's a challenge with so few data, but getting down to functional studies downstream of these findings lends some more credence. So data I didn't show, for example, for ABCC9 and HS are high expression EQTL relationships between the highest hit SNP and ABCC9 expression, for example. So while we don't have the power that we do from 100,000 AD clinical cases, we have to do what we can with autopsy values. But it's a great question.
I had a thought that in the face of heterogeneity, right, if you have highly phenotyped and accurate phenotypes, that is a way out. So just to remind everybody that TMEM106B, that GWAS nobody thought would work, but it was because it was autopsy confirmed that they got they were able to draw homogeneity in there. And so I don't think we need the 100,000 clinical AD level, but if we could build up, we're going to need thousands of these cases to get really good genetics.
Maybe to sort of
I would add that.
Oh. Go ahead.
I would just add that phenotype also matters a lot, like yes or no binary versus ordinal stages versus quantitative, semi-quantitative traits. They have very different power. So we'll have a better bet if we have quantitative phenotypes from large sample, ideally.
I need to follow up on that thought, Hyun-Sik, and again, maybe going back to Eddie. Are we really at the point where we can assemble these cohorts sort of across, the available collections? And the other question I have is of course, the collections have very different designs, right? So some of these more not quite population-based, but at least representative of older communities versus the sort of clinic-based populations. And are we seeing some difference in the genetic associations between these two types of subjects?
Yeah, I think those are great points. And I think the cohort makeup makes a huge difference. Just thinking out loud here, perhaps a thing that could come out of this meeting is the real need for a biological based biomarker, because then you might be able to stratify based on that as opposed to pathology. I think the heterogeneity across cohorts is an issue.
And I feel like despite all those heterogeneity, like TMEM106B, granulin, and to some extent APOE variants were all replicated. The tricky thing is those are already discovered from the GWAS from other diseases and came to latency world. Novel variant discovery in latency, I think, has been more challenging, maybe related to heterogeneity and limited sample size.
Hyun-Sik, actually, one question directed to you. So, you show this sort of nice model where you propose that the APOE interacts with amyloid to affect the latency. Have you been able to translate some of this or this model to some of the data that Risa shared with us, from the HAB study of living individuals? And so do you see, or do you think that some of these genetic variants can help you to resolve some of this heterogeneity of manifestations of people with amyloid proteinopathy that have different levels of cognitive impairment?
That is a very good question. I really want to get to that point, but unfortunately, I haven't been able to get there. Because I think most of these genetic variants, except for APOE, epsilon 4 has a pretty limited sample size, and even like larger, like neuroimaging cohorts, for example, have a limited sample size to actually replicate that single variant association. But hopefully in the future down the road, we may have some polygenic scores coming out from latency genetic studies, and then we'll get to like more detailed phenotypic correlation in living people.
Great. So I think we have 30 seconds. Let me see, there's a quick question here for Hyun-Sik. This is from the audience. Did you look at any ALS associated common variants in your latency GWAS, and UNC13A is mentioned?
Yeah, interestingly, like besides the TMM106B variant, none of the other FTD or ALS associated variants showed up in our study. But still, this is a limited sample size, so let's stay tuned and let's see what it turns out when you have a larger sample size.
Great. Well, thank you again, all three, for terrific talks and a good question answer session. I'll turn it back to you, Pete.
Thank you, Phil.
Thank you very much. Thanks to everybody. Thanks for all the presenters and to discussion leader. And we will now have a 15-minute break and reassemble at 4:45 p.m. Eastern Time. Thank you so much.
Peter, can I just check in the last minute? I'm looking at the chat and I'm looking at the Q&A. There isn't another stream of questions anywhere else, is there?
That's it. That's it. And yeah, so
that's fine.
Technically, you're supposed to sort of concentrate on the Q&A.
Yeah.
And that's supposed to be the one that you should draw from.
Yeah.
And so people that want to ask questions of the panelists should use Q&A. Chat is sort of more of a grab bag. Anybody can say anything to anybody, and I'm sure that there are communications whizzing all over the place between various people. But you can talk to everyone as well if you want to.
Thanks for that clarification.
Thanks for the question.
So before we get started, I know I did it right at 4:45. I apologize.
That's okay.
I believe we are missing one speaker, Dr. Li Wang. If you are on the attendee side and you receive a notification to be promoted as panelist, please accept that and we will promote you to the panelist side. Apologies, Pete.
Great, great. All right, well, it's been a great meeting so far, and I'm very grateful to the team at NIH, including Erica, that's done a fantastic job. So the next round is going to be discuss the topic of epidemiology. The discussion leader is Dr. Brain from Cambridge University, and the first speaker is going to be Dr. Sally Hunter from Cambridge University, talking about TDP-43, granulovacular degeneration and dementia, in the Cambridge City over 75 cohort. Take it away, Dr. Hunter.
Thank you very much, and I really appreciate the invitation to present. I thought I'd race through the first slides, which is really background to the study. So we have the CC75C study design, interview waves every couple of years depending on funding. The brain donations, from survey 2, participants were invited to take part, 242 brain donations, which were processed by the Cambridge Brain Bank according to a modified CIRAD neuropathological protocol, and the diagnostic assessments were done by Cambridge Brain Bank consultant neuropathologists. So CC75C is one of the 3 population-based clinical pathological studies of brain aging in Europe. And these were harmonized, various measures, by Dr. Hanar Kiaj. And past work that we're looking at was by Dr. Suvi Hokkanen, who investigated TDP43 in relation to hippocampal sclerosis and other neuronal loss patterns. And our current work is in collaboration with Professor Steve Wharton from Sheffield University. And we'll be looking at late in the combined donor resource of CC75C and CFAS. So what do we see? Well, I think we've seen the neuropathology already. We don't see many intranuclear inclusions in CC75C, but we do see well-marked GVD, and that's number 3 on the photomicrograph. In comparison to the other studies, CC75C has quite a low prevalence at 27%, but this is based on TDP43 neuronal cytoplasmic inclusions with the phosphorylated antibody. Their presence is associated with age, that's in common with other studies, severe neuronal loss, and severe dementia when adjusted for other pathologies, including plaques, tangles, and neuronal loss. So that means that the NCI are contributing in addition to neuronal loss. We didn't see an association with tangles or neuritic plaques, but this was quite a top-level analysis. So that's Kiaj in 2014. And Hokkanen's results in 2018 and 2020 looked at TDP43 pathology, neuronal loss patterns across the CA1, CA2, throughout the hippocampal formation and hippocampal sclerosis. They're quite nice to look at, papers. When we looked at other pathologies that associated with the antiphosphorylated TDP43 antibody, we found GVD, neurites, glial cytoplasmic inclusions, and neuronal cytoplasmic inclusions. They remain significantly associated with severe dementia when adjusted for age, and also adjusted for the tangles, the neuritic plaques, and the amyloid deposits. We then adjusted additionally for NCI. And when we do that, we found that both the glial and GVD, but not the neurites, retained a contribution to the severe dementia. So we thought, hey, let's look at GVD in more detail. It's a very, well, relatively disregarded pathology. But we also have to be careful because it could, there's an association with TDP43 with stress granules. And when we think about this, we've got granular TDP43 cytoplasmic inclusions. We've got stress granules. We heard mention earlier TDP43 granular pre-inclusions. And we also have GVD. And I'd like to ask the question, are we adequately distinguishing between these granular type pathologies, or are we not adequately controlling for them? There are 2 different perspectives for the contribution of GVD. Could it contribute to a pre-inclusion, or does it represent another pathway for the contributions of TDP43? And how does this fit in with what else we know about GVD, which was brilliantly reviewed in Colo in 2016, including the late lysosomal system markers, unfolded protein response markers, cellular stress, and tau. And if we think about that, that was raised earlier by Lisa Spielman, the contributions of wider protein homeostasis. And I think GVD could fit here. Oh, I just thought I'd remind myself that when we talk about pre-inclusions, we have to be careful about implying a progression from cross-sectional evidence. We also did work with tau, where we see very similarities in approach in relation to GVD. Does GVD represent a different pathway for tau aggregation, or does it represent a pretangle? And if it represents a pretangle, how do we make sense of the association with TDP43? So I'd like to acknowledge our participants, their families, the NHS, prior work by Subi, Hannah, Thais Manette, Dr. Tuomo Polbakowski, Dr. Jane Fleming, and Professor Carol Brain and Dr. Richard Merrick, the Cambridge Brain Bank Laboratory that hosts our resource, and our funders. And I'd like to leave you with those questions, which I've covered the first three, but I'd like to ask, does GBD involve the same components in all cell types? Is it a gateway pathology? And should we be paying more attention to it in our study designs? Thank you.
Thank you kindly, Dr. Hunter. Great talk. And the next person is going to be Dr. Aaron Abner from the University of Kentucky, talking about late NC and multiple proteinopathy in a community-based cohort of brain aging, results from the University of Kentucky Alzheimer's Disease Research Center. You're on,
Thank you, Dr. Nelson. Apologies for the long title there. So I'm just going to take credit for presenting this today. This is a team effort, as are most of the things that we do. So this is based on a paper that we published in June of 2020 in JAMA Neurology. It was led by Dr. Kieran, who was my PhD student, and she is now a postdoc at the University of Florida. And you can see lots of our friends here on the author list. So I want to orient you by saying this is a study of our autopsy participants at the UK ADRC. We needed to have data on TDP43 to do this, as well as some other things. And we required for this analysis that everybody had at least some tau pathology, which, as we've already heard today, is basically everyone when we get into late life. So we have 375 autopsy participants here, and they die about age 87, which again is consistent with a lot of the other data that we've heard today. And what we did was we categorized all of the participants by the 4 proteinopathies that we've been talking about all day: tau, amyloid, alpha-synuclein, and of course TDP43. And this work sort of arose out of an observation that Dr. Nelson made many years ago, that we started to have some participants who had all 4 of these pathologies, and it was really striking. And it seemed like this might be rare or something that was explained by a particular genetic signature. But we soon realized that this was actually really common, that people would have all 4 of the pathologies. So for us, 46 out of that 375 have all 4 of those pathologies. But we also have 24 of those people who only have tau and TDP43. And we had 71 people with tau, A beta, and TDP43, but no alpha-synuclein. So overall, about 38% of all of our autopsy participants in the study with tau also have TDP43. These arrows down here are just to orient you on the slides to where the TDP groups are. I also want to point out quickly on this slide that there is a progression of cognitive impairment that we see as we move from the left to the right across the screen, and the people with all 4 or with 3, including TDP43, have quite severe impairment at the end of life. And the people with all 4 all died either with dementia or with normal cognition. And it turns out that those people who died with normal cognition in QMP or tau and TDP who do okay, they have Brock stages that fall in the 1, 2, 3, 4 range. I want to point out the time and state, which is really striking to us. So overall in the sample, about 3 years in MCI and about 5.5 years in dementia. Alzheimer's disease, as we would normally think of it, probably very similar to that, maybe a bit shorter in the dementia phase. But we were really struck by our participants that had all 4 of those proteinopathies. They had a really swift course of MCI, went right through it into dementia. And so that also explains that normal and dementia at the end there. When we looked at people who died with dementia or who died with MCI and categorized them again by these proteinopathies, we found that 19% of our participants with dementia had all 4 of those proteinopathies. So again, very common. 27% of them had tau, A beta, and TDP-43. Just 5% of our dementia cases were the tau plus TDP-43. And I do want to mention, there was a comment earlier about, do you see A beta? I think it was Risa, do you see A beta and TDP-43 just by themselves? In our cohort, no, we hardly, I don't know if we see any cases like that. This study, we don't, because we required tau, but we've been doing some more recent work and we don't see it. So there is something interesting there. Also towards the clinical course, I want to draw your attention to these figures, which are showing the predicted probability of obtaining either a normal or a severely impaired MMSE score in the 12 years before death. And we find that, as has been suggested earlier, the people who have TDP-43 plus tau and A beta, which is the green line, and then if we add alpha-synuclein, this orange line, they are already having trouble, right? A decade before the end. They have the lowest probability of having a normal MMSE score. The blue line here that I'm pointing to is the tau plus TDP-43. So they are having some cognitive impairment, not quite as severe if we don't have A beta and alpha-synuclein. And again, on the right here, we have the probability of obtaining a severely impaired MMSE score, and the QMP group having all 3 is always right there at the top. And then tau, A beta, and TDP-43 is right behind. So these are really impactful proteins that we've heard about, and they're very, very common. Again, they're very common. So these are our hypotheses about what the clinical course of these diseases might look like if the experience of our participants were truer broadly. So we've got the orange line here, which is our all 4 pathologies. And again, this reflects a very early impairment in a swift course, followed by hanging out for quite a long time, many years, severely impaired, which suggests to us that the accumulation of all 4 of those pathologies could well be happening much earlier in the process, as opposed to, for example, starting with perhaps A beta, accumulating tau, and then later getting alpha-synuclein or TDP-43, and then the other one, suggesting that happens much closer together in time and much farther ahead of death. If we have tau only, we're okay, but anytime we add TDP-43, we have a big hit to cognition. And as we've mentioned, we don't know a lot else about it, but we are starting to work on neuropsychiatric symptoms that may be associated also with these pathologies. So just quickly, it's really common, it's really bad for our cognition, and so we have to figure out a way to include it in clinical trials or account for it, because it's going to keep causing problems, I think. So to sum up, we are profoundly grateful to our participants and their families, without whom none of this could be done. This particular study was funded by the UK ADRC P30 grant. Our former number is crossed out here in yellow. We have the new grant number. So if you use our grant number for anything, please update it, as well as an R01 led by Dick Criscio, myself, and Leiu from Rush University. Thanks for your attention. I'll stop sharing.
Thank you very much, Dr. Abner. Great talk. Next speaker is Dr. Maria Carrada from the University of California, Irvine. Title of her talk is Latency in Dementia and the Oldest Old Findings from the 90+ Study. Thank you, Dr. Carrada.
Sorry, forgot to unmute myself. Thank you to the organizer for the opportunity to present here today. We have seen late neuropathological changes occur more often at advanced ages. So we wanted to explore the relation to dementia and to other neuropathologies in a cohort of oldest old individuals. I first wanted to, I have nothing to disclose, but I wanted to acknowledge our funding sources and collaborators, in particular, Claudia Kawas, Ahmad Sajadi, Ron Kim, and Tom Antin. And very important to us, John Trzajanowski, he was a collaborator of ours for many years. And he actually started the TDP-43 investigations in our cohort. So much of what we have learned that we're doing now here is thanks to him. I will be describing findings from 401 participants in the 90+ study. which is a community-based autopsy cohort of people 90 and older in Southern California that began in 2003. The average age at death is 97, 69% are women, relatively highly educated, and just under half had dementia at the time of death. We started with the prevalence. In this cohort, about 36% have late neuropathological changes in all 3 stages, and 1 or higher. And we have the highest prevalence being stage 2, which are TDP-43 inclusions in the hippocampus. The prevalence is higher in centenarians than nonagenarians. We do not see a difference by sex, but although we don't have many individuals in stage 3, they seem to be disproportionately women. We don't have any association between latency prevalence and APOE4 or APOE2. What about dementia? We have an increasing prevalence of late NC with higher degree of cognitive impairment, with late positivity in 50% of people with dementia. On the right side, you see a graph of the odds ratio of dementia in relation to late stages. And even at stage 1, we actually see, which are TDP-43 inclusions in the amygdala, we see that although that it's a low prevalence, it is still strongly associated with dementia, with an odds ratio of about 3. And so given the similar association of stages 1 and above with dementia, for further analyses, we justify the dichotomization of late positivity as stages 1 or higher. With that, we see here that individuals with late NC positivity are 3.5 times more likely to die with dementia than those without late NC. So a fairly strong association between late and dementia. So we also wanted to explore the association with late neuropathological changes in relation to other neuropathologies. So starting with hippocampal sclerosis, we see that 65% of people with HS are late positive. So about 1/3 of hippocampal sclerosis is not related to latency in this cohort. This gives us a very strong association between hippocampal sclerosis and late, where individuals with hippocampal sclerosis are almost 4 times more likely to be late positive. What about neuropathological, AD neuropathological changes? With neurofibrillary tangles, we see a very clear dose effect where we have increasing late positivity with increasing Bragg stages. Another way to look at this is to look at the increasing odds of late positivity with increasing Bragg stage. For measures of amyloid, we do have an association with serial plaques, and we see similar odds ratios for moderate and frequent plaques. We don't see a clear association with Thal phases. I do want to point out that overall we see that although it is less likely, it is not infrequent to have late NC when we have no or minimal Alzheimer's disease neuropathology. We already saw that there was a strong association of late NC with dementia. So now we wanted to explore if the association was independent of other neuropathologies. In a logistic regression model that included 8 different neuropathologies, we see that late NC has a strong association with dementia, an odds ratio above 3, and that is higher than that of Alzheimer's disease neuropathological changes and only lower in magnitude to microvascular lesions. And the association is independent from all the other neuropathologies. So using this information, we applied methodology that was used earlier by a collaborator, Ron Brookmeyer, and more recently by Dr. Patricia Boyle, as Dr. Nelson showed earlier, to estimate the proportion of dementia that can be attributed to the different neuropathologies. And thank you, Dr. Boyle, for sharing the code that allowed us to implement this. We see that a significant proportion of dementia in the oldest old, about 20%, can be attributed to late neuropathological changes. And a proportion that in this cohort is not much lower than that for Alzheimer's disease. So a very important neuropathology in the oldest old. And in summary, in the oldest old, we see a prevalence of latency about 36%. It is more common in centenarians than nonagenarians. We see a strong association of all stages of late NC with dementia, a strong association with AD neuropathologies, in particular neurofibrillary tangles and neuritic plaques. About 2/3 of hippocampal sclerosis is late positive. We also noted that the association of late neuropathological changes with dementia is independent of other neuropathologies, including Alzheimer's disease. In terms of public health impact, about 20% of dementia cases can be attributed to latency, a fraction that is not very different from AD at very advanced ages. And I finally want to give a plug for a poster by my colleague Amal Sajadi of work that shows distinct patterns of cognitive decline in latency versus AD and C in this cohort. So with that, I'll stop sharing my screen. Thank you.
Thank you very much, Dr. Kurata. And with that, we'll turn over to discussion leader, Dr. Carol Brain.
Thanks very much, Peter, and thank you for organising this amazing meeting and all the team that's led to it coming together so nicely, such a stimulating meeting. And thank you to all the speakers for being very timely as well. We have a few questions. I'm going to start with, they keep disappearing actually from my screen. I'm going to start with one for Sally, which is actually in the chat, and it's about necroptosis. So GVD having been shown to be related. So Dietmar Tal has raised this and also given a paper. I just wondered whether you wanted to comment on that, because you really set out a provocation, and I think there will be others who want to respond.
Yes, and I think that the situation with GVD is not clear because no one's really focused on it particularly. So the necroptosis, the cell death, it does seem to be that GVD is a gateway pathology between different pathways or different components of the protein homeostasis system, and that in itself, if that falls over, that will lead to cell death. That's not a surprise. I think the comparison that maybe Dr. Tal's asking is looking at GVD having an association with cell death, whereas TDP43 inclusions or pre-inclusions aren't necessarily associated with cell death. I think it's quite difficult, because this is a cross-section, to know whether cells have died and been cleared away, especially when TDP43 is actually associated with neuronal loss. We don't know whereabouts in that pathway we've landed when we're looking at a section. So I think more work needs to be done.
Yeah, so I suppose that comes back to what's taken out from this meeting on, which is how to think about the cross-sectional aspect of pathology that is looked at and the characterization, and that's something that, Peter, I'm sure you'll be thinking about carefully, and the relationship to additional pathologies. So thanks, Sally. I wanted to come on to the role of vascular pathology, and I'll turn to Erin and Maria, I think, just to maybe reflect on that, because you've both got vascular pathology within your cohorts, and very carefully characterized and sometimes separated out. Maria, you showed the independent effects in yours. But the fact is that all these pathologies are interrelated, and really throughout today's conference, vascular has been relatively neglected, and I would just like you to comment on that, given the fact that in population cohort studies we see a lot of vascular pathology. Erin, do you want to start off and then go to Maria?
Sure, yeah, absolutely. Vascular pathology is really important, and one of our co-authors on that paper, Dr. Katsumata, is moving in to incorporate those pathologies into this sort of model of the universe. In our cohort, we don't see that much vascular dementia, and the things that we see a lot of, like brain arteriolosclerosis, don't tend to be super strongly associated with cognitive impairment, although they certainly are. But we haven't noticed yet that they tend to associate with a specific neurodegenerative phenotype, for example. And so our assumption is sort of that the vascular would be affecting all of those groups in the background in roughly the same way. But we'll find out.
Yeah, absolutely. Yeah, and Maria?
Yes, I think one thing that I wanted to point out is that in our cohort, when we talk about vascular pathologies, it's usually not large infarcts. People at this age don't die usually, or we don't see large infarcts very often at this age. So what we actually see is just microvascular lesions. And actually one thing that was very interesting that we actually noticed very recently is that atherosclerosis, it's very common. We knew it was common, but we were always, when we were looking at it, we were always dichotomizing as moderate and severe versus mild and absent. And we never really saw an association. And we started noticing that even at mild levels, atherosclerosis is actually very also strongly related to dementia. So now once we actually put atherosclerosis in that level, we actually see a very high prevalence, very strongly related to dementia. So it seems like to be more of these sort of small vessel type lesions that are actually more related in our cohort. We haven't yet looked at it in relation to decline per se, but as you saw, I mean, when we combine all the vascular neuropathologies, they actually account for about 40% of dementia in our cohort. So it is definitely a very strong contribution at this very advanced ages.
Yeah, thanks. And I would just like to come back to that, the fact that the average age of death is about around 87, actually similar in CC75C and the ACT, and then of course it's older in yours, Maria, and the fact that we're talking about trials and so on. None of the trials that are done are in those age groups. So I'd just like you to reflect on the importance of age, and perhaps ask you, I think we've got a couple of minutes, maybe think about really the age has underpinned pretty well every presentation. And I think it's really important that we reflect on this, particularly from the cohorts that are represented in your talks. So Maria, I'll start with you with the oldest old.
I think one thing that's very important is, I mean, dementia is very prevalent at this age. So when we're actually talking about dementia in the population, a very high proportion of them are at this very high advanced ages. So not including oldest old in studies, in trials, it's neglecting a very high proportion of people that are actually affected by this condition. So I know that it's complicated because what we're seeing is at this very advanced ages, we have a large combination of pathologies that basically they have a lot of different things. So it's much harder. Clinical trials usually try to be very clean, but we are neglecting a very, very, very big, large subset of the population that actually are impacted the most by dementia.
Thanks, and Erin?
I would just say quickly that the pathologies that we study, we know develop and exist in the brain for a long time, right? Prior to when we see them at autopsy. So if as our figures were showing, you know, our average age of death is something like 87, they start to have impact from these declines in the mid 70s then, right? And so they would be in the age range of people who are getting recruited into clinical trials. So I do think it's highly relevant.
Yeah, okay, thanks. There are some more questions coming up in the chat now, but I don't think we've got time to answer more than one of them. Maybe just Sally, you could just answer this one. A similar pathology to late ever seen in young patients? Is there something about the older brain that predisposes to this phenotype?
I don't know. I've never looked at any brain that's younger than 75.
Others might be.
I can't. That's not my. It's not me.
Yeah. Okay. Well, lovely. Thank you so much for all of you for your lovely talks, and we'll move on to the next section. So back to Peter.
Thank you very kindly, Dr. Brain, and all the panelists. That was wonderful discussion, and I highly encourage people to keep up discussion with everybody that has already talked. A great thing about the webinar format is that you can keep up the Q&A, you can keep up the chat, you can keep up talking, and we will now, however, keep going forward here. Start with biomarkers. Dr. Arfanakis from Rush University is going to be the discussion leader. And our first talker is going to be Dr. Lei Wang from the Ohio State University. And the title of his talk is Unique Contribution of TDP-43 to Antemortem Hippocampal Shape Morphometry. The floor is yours, Dr. Wang.
Right. Thank you, Pete. Thanks for the organizers for the invitation. Give us a chance to talk about our recent findings on the hippocampus. So everybody, everyone has heard the challenges in seeking biomarkers for TDP-43 or late. The challenges, of course, include multiple comorbid pathologies and also substantial inter-individual variation in the neuropathological phenotypes. And there has been some pioneering work from the Rush group that looked at how postmortem imaging could be related to postmortem neuropathology. So in this study, their group has showed that amygdala shape can be found to be associated with specific patterns of TDP-43 staging. And of note is the finding that spatial patterns of associations between TDP-43 and AD pathology are visually different. And so our work kind of built on this concept of looking for shape variations in brain structures known to be affected by TDP-43 and see how they can be associated with TDP-43 pathology and whether they can be different from other pathologies. And since our goal is to look for neuroimaging biomarkers, we thought to look at antemortem imaging data versus postmortem imaging. So this is current study that we're reporting to as a recent data that's under review. And we used the cohort from the Rush-Rossmap studies. And well, we wanted to include subjects that had antemortem imaging, MRI, closest to their autopsy, and those who have neuropathological data. We included 99 subjects, and here's the demographic information. And as you can see that they all had, most of them had more than one type of pathology that we looked at our TDP-43 amyloid and tangles. Okay, and so the way the TDP-43 quantification was performed was that the inclusions were looked for in 5 different brain regions: amygdala, hippocampus, CA1, and dentate gyrus, as well as entorhinal cortex and the midfrontal cortex. And we summarized the quantitative ratings from these measurements and turned them into a semi-quantitative rating in all the regions. And then we summarize across the 5 brain regions to come up with a global TDP-43 severity that we use that to represent each individual and associate with neuroimaging data. And here's a histogram of that global TDP-43 severity across all the individuals here. And for mapping of the hippocampus, we used an algorithm that we've developed for the past few years, and used a multiple atlas library of 74 scans with expert manual segmentations of the hippocampus. And that creates a unbiased mapping of the hippocampus in each individual. And we performed surface analysis that relates surface variations to the postmortem TDP, global TDP severity, while accounting for global A beta, tau, as well, some of the demographics, interval between MRI and death, as well as comorbid pathologies. And when we looked at the relationship with TDP-43, we excluded the hippocampal sclerosis that were found in the 5 individuals. And to correct for multiple comparisons, we used a surface-based random field theory with a family-wide error rate of 0.05. So our major finding is that for TDP-43, we found that elevated TDP-43 global measure was related to inward surface variation, as indicated by blue colors on the screen here. And it seemed to follow specific patterns for the medial side of the spiculum, as compared to the patterns of A beta in this cohort that did show a visually distinct pattern that were following mostly in the CA1 subfield, as well as the spiculum. And when we summarize these regions that are associated with the TDP-43 and use the surface variation data to relate to cognition, we found that they broadly related to the decline in memory and other cognitive domains across these different domains. So to summarize, we found that there are distinct patterns of association between TDP-43 and antemortem hippocampal surface. And that corroborates patterns of association found from ex vivo amygdala data in the way that the associations are different between pathological entities. And we also found that the surface deformation that are associated with the pathology was also related to reduced cognition. So to go from here, since we're looking for neuroimaging biomarkers, I think then the future directions would include analyzing the patterns across the regions known to be affected by TDP43, as well as white matter regions that connect from these regions. And we also would like to see whether these patterns can be found earlier in the time possible that can be detected in the search for a possible signature of mixed pathologies. So with that, I'd like to acknowledge the participants and our Rush collaborators, and give a shout out to my graduate student, Ashley, and especially the NIA Diversity Supplement directly funded this work. Thank you.
Thanks, Dr. Wong. Great job. The next speaker is Dr. Robert Rissman from the University of California, San Diego, and the title of the talk is High Throughput Plasma Bioassays for Late. It's all yours, Rob.
Guys, everyone, thank you, Pete. Thank you, Nina and Julie, for the invitation here today. I'm going to talk to you a little bit about some recent data we have from the lab, where we are characterizing a new bioassay for plasma detecting TDP43. I don't have any financial conflicts of interest. I don't have any funding related to late or TDP43. And I've listed a couple other things here that you may or may not be interested in. My lab is predominantly funded by federal and private grants. And I'm a leader for a couple of different organizations, Service Corps, ADRC and ACTC, ADCS. So the approach in our lab is to not only look at plasma-based biomarkers in neat plasma, but also to look at it in extracellular vesicles, such as exosomes. And why would we consider looking at exosomes? Well, as you can see here by the schematic, TDP43 is one of many proteins that is circulated in the brain and is passed from one cell to another via exosomes. So you can see here either by exosome trafficking or other extracellular vesicle trafficking, TDP43 has been observed in a number of different circumstances and in a number of different diseases. So in coming out of the original late meeting, which I was involved in, I realized that we definitely need some sort of way of being able to detect these participants ante-mortem. As we saw from Dr. Wang's talk, he's working a lot on neuroimaging. I started to think a little bit about what bioassays we could use for these things and how we could differentiate these late participants from others with other neuropathologies. So what we did is we started speaking to Pete and others about potential samples we could get to take a look at. University of Kentucky has a great number of these plasma samples from late donors. And as you can see here, there's sort of a layout of the samples that we received from Dr. N. Some of the participants have no late neuropathology. Those are the ones there on the left, NC negative. And the NC positive were people who did have late pathology. As you look through kind of the little small short demographics that I put here, you can see that the majority of people in both groups are demented, some more than others, and some of the other factors that you see in here. One thing, of course, that's missing is diversity in this cohort. The majority of the donors were white. So again, this is something we're going to look to expand upon later in our studies. So what else did we use? Well, there are very limited numbers of assays that exist commercially for TDP43. So we thought, we're going to have to try to make our own. So I dropped John Trojanowski a line, and indeed he told me at that time that he had multiple antibodies that he could send me and that we could test. So here's the list of all the different antibodies he sent. I don't know how familiar you guys are with them. I believe they're all published at this time. So we selected one of them and decided to try that one first and see how it went. Still, the other ones we still have, and we are planning to consider testing. So what do we actually do with these plasma samples when we make our ELISA? So we have published methods for isolating exosomes from human plasma and differentiating different types of exosomes, such as neuronal exosomes, astrocyte exosomes, microglial exosomes. And again, a lot of those data are already published already. To make this new ELISA, we used the antibody we received from Penn, and we also used just a commercially available detection antibody to actually read out the assay. And sort of the methods are kind of laid out here just very briefly. I'm happy to expand upon these in another session or offline if you guys would like. So when we load up our exosomes, we purify them and we make sure that they are what they think they are. We use FACS sorting, and we often use nanocyte as well. You can see here on the left in these panels how we identified the neuronal exosomes using L1CAM, GLAST for astrocyte exosomes, and TMEM119 for our microglial exosomes. And then before we run any bioassays, we always make sure that we have approximately the same number of exosomes in our pools, and we assay the same number of exosomes in our plates, right? So we don't have more exosomes from neurons than others, and things all the same are loaded onto each plate. So how did the data look? Well, I could tell you, very surprising. I hadn't expected such dramatic changes. I had thought perhaps there would be changes in neuronal exosomes and perhaps not some of the others. But comparing the commercially available kit, which you can see here is from Casubio, we saw some very dramatic changes in astrocyte-derived exosomes. This was even further sensitive in our homemade assay. As you can see there on the y-axis, we can detect a whole lot more TDP43 from these astrocyte exosomes. There was practically no TDP43 in astrocyte exosomes from participants, from donors, who did not have late neuropathologic changes. Really, I mean, so Charisse Winston is the postdoc who did the majority of this work. And literally, when we first looked at the data together, there was nothing in the late NC bar graph area. And I'm like, Charisse, what's going on here? She's like, well, they had nothing. So anyway, just to note that we actually saw nothing, I asked her to put a whole bunch of data points in there at the bottom. I believe there was really only 4 donors that had any detectable level of TDP43 and the negative neuropathologic changes. So we saw not a whole lot going on in the neuronal exosomes. Those levels are approximately the same, although again, with our homemade assay, we're able to detect a whole lot more of it. In terms of microglial exosomes, you see here the data is very confusing. So they're going in 2 different directions depending on the assay that you use. So with the homemade assay, you can see that we're able to detect more TDP43 in the microglial exosomes from the neuropathic changes consistent with late. But in the commercial assay from Casubio, it's kind of mixed. It's almost the other way. We're unable to really understand why that is, and we don't have a whole lot of information about that antibody that they're using in the commercial kit. It's just not available. It's proprietary. And that's why I'm kind of excited that we kind of made our own and we're able to understand all the differences in it. So anyway, very interesting changes in astrocyte exosomes, but not necessarily in our other exosome pools. So what about in just straight plasma? Well, again, in our lab, we often just assume that we want to look in exosomes first and see what's going on in those, and then perhaps look in neat plasma. Here, we didn't quite have enough plasma to look in unmanipulated plasma, but this is the exosome depleted plasma. So this is what's left over after Dr. Winston took out all of the exosomes. So you can see here that there's really very little difference in the amount of TDP43 that's sort of floating around in plasma that was not tied up in exosomes. The levels are relatively high, but yet there's no significant changes. There was a lot of inter-subject variability for these. So kind of to sum up here, we're able to detect TDP43 in all kinds of exosomes, neuronal, microglial, and we're able to see this in both types of donors, in negative neuropath change donors and positive. However, in astrocyte-derived exosomes, we can really differentiate people who have late pathology and who do not have late pathology. Left over in the plasma, we do see levels of TDP-43, but we don't see a major difference between the two groups. Where are we going from here? Well, I think we need to further, further specify and characterize our assay. Again, these data are very new. I think I'd like to look in a few more donors from Pete and hopefully from our ADRC at UCSD and really see the dynamics of this assay. I think in the meantime, we're kind of preparing to write up these data and share them with the general community, and we'll see how that's received. I think also another important thing we're going to need to do is look at actual just neat plasma samples. Again, unmanipulated plasma that has not had the exosomes yet removed from them. But anyway, I thought these data were really exciting and they were really unexpected. So I just want to thank the people who actually did the work, as some of you are. I just sit at a desk and type all day and take phone calls. We really need to thank Charisse Winston, who is a K99R00 recipient. She did all of the work for this project, and she did it really efficiently and greatly. I just want to mention also that she is, with her K99R00, she's heading out to look for a faculty position now. So if you have availability, please drop her an email or drop me an email. Of course, we wanted to retain her at UCSD, but I'm not sure that's going to be possible with our current resources. So kind of a big opportunity for people, I think. I know I'll miss her when she goes. I also want to thank, of course, Pete Nelson, who's a great collaborator. Thank you, Pete, for sending all these samples over. And I'm going to greatly miss Dr. Trojanowski, who's been a friend and a colleague for over 20 years and for sending me these antibodies. Thank you, John, wherever you are. Much appreciated. And thank you, everyone, for your attention.
Thanks kindly, Dr. Rissman. That is very fascinating work. And the next speaker is Dr. Michelle Grothi. Apologize if I mispronounced. The title of the talk is In Vivo Stratification of Amnestic Dementia Patients based on an FTG PET signature of autopsy confirmed late NC. The floor is yours, Michelle.
Yeah, hi. Thank you so much for inviting me to present here at this wonderful meeting. And I'd like to start with a brief summary of some of the best established clinical features associated with late that we have also heard a lot about today. And this is, of course, advanced age, somewhat less aggressive disease course generally, with a more memory selective cognitive deficit. And then there are some specific and well-established genetic features, such as enrichment for the TMM106B risk allele and a somewhat lower frequency of the APOE4 allele when compared to AD. And as we have already seen this morning, late and hippocampal sclerosis also appears to associate with distinct temporal limbic distribution pattern of hypermetabolism on FDG PET. And in the current study, we wanted to further explore the potential clinical utility that this pattern may have for differential dementia diagnosis. So for that, we first wanted to reassess the late associated FDG PET pattern. And we used data from the Autopsy cohort, which currently includes 50 autopsy cases who had clinical diagnosis of AD dementia or AMCI at last clinical evaluation and an antemortem FDG PET scan. And of these, we identified 7 cases who had limbic TDP-43 pathology, but no or low AD pathology, and 3 of those also had hippocampal sclerosis. Another 23 cases had typical AD pathology without comorbid TDP-43. And now when looking at the contrast to healthy controls, we see that the pathologic AD group indeed shows this highly AD typical pattern of temporal parietal hypometabolism. But in contrast, the late group shows a very distinct pattern, which is characterized by much more pronounced involvement of these temporal limbic areas. compared to the AD pattern. And this pretty much resembles the pattern that we've seen this morning and also on this previous slide. Let me see if I can move that. Okay, so in a direct contrast between the two pathologic groups, highest effect sizes were observed for more pronounced hypermetabolism in late in the medial temporal lobe and in inferior temporal and lateral parietal areas in AD. I think this was okay. So we then wanted to move on and further explore the potential clinical utility that these autopsy-derived patterns may have for in vivo patient stratification in a separate cohort of about 250 patients who were clinically diagnosed with AD dementia. And for that, we assessed the spatial correspondence between each individual's hypermetabolic C-score map with each of the two pathology-specific patterns. And using this approach, we indeed identified, sorry, I think this, okay, here we are. We indeed identified a subset of patients that showed a hypermetabolic pattern that was much more similar to the late pattern than to the AD pattern. And this was the case for about 10% of the patients in this cohort. Not surprisingly, a much larger portion of patients showed a match to a classic AD typical pattern of hypermetabolism. So these are the average hypermetabolic patterns of the stratified late-like and AD-like patient groups. And I think this is a pretty nice sanity check that the automated pattern matching algorithm actually does a pretty good job at picking out these or detecting our patterns of interest. Sorry. So now when looking at some of the clinical features of patients with a late-like hypermetabolic pattern, a first striking finding is that these were on average 10 years older than the AD typical cases. And they also had a more memory selective cognitive deficit as indicated by this cognitive profile variable here, which quantifies the difference between composite scores of memory and executive function. And these patient groups did not differ in dementia severity at baseline, but the late-like group did show a considerably slower course of cognitive decline over follow-up. And finally, CSF biomarkers confirmed significantly lower levels of AD pathology in these patients, both for amyloid and for tau. And the late-like patients also had a lower APOE4 allele load with a complete lack of homozygous carriers of the APOE4 allele. By contrast, the late-like patients showed a higher frequency of the TMM106B risk allele and with a complete lack of the protective C allele. So in summary, our findings corroborate that late associates with a distinct temporal limbic pattern of hypermetabolism that clearly differs from the atypical pattern of temporal parietal hypermetabolism. And this pattern is provides clinical utility for differential dementia diagnosis because clinically diagnosed AD dementia patients exhibiting this pattern had significantly lower AD biomarker levels and showed specific clinical and genetic features that would be consistent with underlying late neuropathology. So with that, I'd like to thank all my colleagues here at the EBIS in Seville and my international collaborators on this project. Thank you very much.
Thank you very kindly, very nice talk. And with that, I turn it over to Dr. Arfanakis for discussion.
Thank you, Pete. It has been great all day today. I'm glad the you kept the biomarkers at the end so that you kept the crowd around waiting for the good stuff. So let me give a very brief summary, and then we'll go on to a few questions that we have here. So we all realize that biomarkers are important. We're looking at it from the PET view, from the MRI view point of view, and also from the blood-based biomarkers point of view. This will help us understand the disease better during life. It will help us select participants for clinical trials, not only clinical trials for late, but also clinical trials for AD. I think this is very important. Yeah, so this will push forward treatments and the research for latency. So let's go over to a few questions here. There is a question from Binzu to Dr. Risman. He's asking if you know if the antibodies you use from UPenn are anti-TDP43 or anti-phosphor TDP43 at different sequence segments. Dr. Risman.
Yes, I'm here. Though I think those are anti-TDP43, not necessarily phospho. I think they're phospho blind. They could be pick up both forms.
Okay, thank you for that. We have a couple of questions for Michelle. In terms of the patterns that you see with FDG PET, when you have late with and without HS, and when you have late with and without AD, have you looked into that?
So we only had 4 late cases without hippocampal sclerosis and 3 that had hippocampal sclerosis. I have to say, so I'm not a neuropathologist, and I don't know exactly how hippocampal sclerosis is diagnosed pathologically, but so I wondered a bit about this low proportion of hippocampal sclerosis cases in these 7 individuals, because on MRI, all of them have severe medial temporal lobe atrophy, and all of them are by definition Bragg stage below 4. Actually, there was only one with Bragg stage 4, and all the others had Bragg stage 1 or 2. And so I don't know if you equal hippocampal atrophy on MRI to neuronal loss, I don't know if you can do that, then that would kind of meet the definition. So I don't know why in these other 4 cases no hippocampal sclerosis was diagnosed. But yeah, so the answer is I couldn't assess this difference in this small cohort. So in this individual basis, they all pretty much show this pattern in their individual CMAPs. So that I don't think there are big differences. More interesting probably is the question whether there's differences between those with comorbid AD pathology. And so we had several of them in this cohort. I didn't show them here for sake of time. And interestingly, they did not differ at all from the pure AD cases. And that was kind of unexpected. There were 2 cases with combined AD and late pathology who had hippocampal sclerosis, and both of them showed were pattern matched to the late typical pattern. But since there was so much variability in this mixed, in this comorbid group, there was no significant group differences. And this may be related, that's kind of different to what others have found. And I think that may be explained by the fact that all of these AD cases had ADNC of 3. So full-blown AD pathology. And the additional effect of TDP43 in AD may be more pronounced if you have like lower levels of AD pathology, which there weren't in this cohort.
If I can also ask a question here, could you comment about the pattern that you see with FDG PET for latency, and also the pattern that has been published, the atrophy pattern that has been published using MRI?
Yeah, I think we also looked at the atrophy pattern on MRI in this cohort, because all of these cases also had antemortem MRI scans. And in general, I'd say it's similar, but less pronounced. And so you have this atrophy pattern. Atrophy on MRI is much more specific to really the hippocampus and the medial temporal lobe. And the FDG pattern seems to extend a bit more. I don't know whether these are local effects or maybe some remote effects of some functional things that are being picked up in these FDG PET scans. It generally works if you take this, if you look at the patterns and then the inferior to medial temporal lobe ratio that had previously been proposed really does a very good job in separating these two groups. Basically, this whole brain pattern comes down to, can be summarized quite nicely in this simplified ratio. And if you calculate the same ratio for MRI, it also separates your group, but at a lower effect size.
Okay, thank you very much. The data that Robert, Michelle, and Lei showed us are excellent, and there's a lot of work that needs to be done for biomarkers. Pete is sending me messages. We need to wind it down. Pete, the floor is back to you.
Okay, well, I will get, I think that I'm supposed to give the concluding remarks. Julie, do you have something that you'd like to say penultimately?
Just thank you to everybody who hung in there. We still have over 175 people online, and this just goes to show the interest in late and appropriately so. It's super important, as noted by the epidemiologists, and yeah, and great job to everybody today.
Well, I agree, and so I call you guys the long haulers, you 173 suckers. And so, but one thing I do want to say is that this has been a wonderful meeting. I want to conclude by sort of saying that maybe we should be inspired by Dr. Trojanowski in another way. And the thing that I was thinking about is he's one of the very few people that has active feet in this different camps of the dementia world. He was a clinic-based person with FTD, but he worked with Claudia Kawas and their crew to do an epi sort of community stuff. And he worked a lot with patients and the clinicians as well. And so bridging those bonds and getting together, and sometimes we know that this late and FTLD worlds has had this sort of camp mentality. The points that were made and raised by Dr. Dixon about the need to generate this sort of these borderline between FTLD and HS and late, and the points that Manuela made that we need to tweak our criteria. Those are great points, and we need to get together to solve these things and work together, informed and inspired by people like John. And I think that that's a good way of thinking that this, hopefully that this conference will help move us in those directions. So thank you so much to everybody for coming. Thanks to all for staying, and bid you adieu.
Thank you, Pete. Very well said.
Thanks, everyone.
Thanks, Bob. Bye, everybody.